honored to speak to our speaker today, Dr. Chip Taylor, who is the Emeritus Professor at the University of Kansas and the founder and former Executive Director of Monarch Watch. Dr. Taylor got his training in insect ecology at the University of Connecticut. He not only is an expert on monarch butterflies, but at the height of the killer bee movement in the 70s and 80s, when they even got on Saturday Night Live, he did studies around that. So there's seldom an opportunity that I have to talk about people who've had, major impacts, beyond everyone wants to make an impact. There's very few of us that make an impact on a state or even local level. There's even fewer that make an impact at a global level. And Dr. Taylor has done that through his work with Monarch Watch and the conservation education work that they do that is now global. He's going to speak to some of the success that they've had, not only in tagging and tracking monarch butterflies, but in the work that the master gardeners are so involved with, the creation of the Monarch Way Station program. The very first one here is in Lawrence, Kansas. Those of you who are online understand that we tour our demonstration gardens, including Monarch Watch. And if you're interested, you or your group are interested in touring the demonstration facilities, we'd be happy to do that. But I think the best person to learn more about Monarch Watch and the Way Station project is Dr. Chip Taylor.
Better get hooked up here. I'm sure you can all hear me anyway. All right. Well, good morning to you all. I'm pleased to be here. I'm pleased to see a couple of the new Monarch shirts. Thank you for wearing those. That I had nothing to do with that shirt. I designed the first Monarch T-shirt. It had butterflies coming out of your pants in the spring, and on the back it was going into your pants. It's to symbolize the migrations both ways, right? and of course the butterflies got bigger as they got further north. Anyway, that was my T-shirt design episode of life. Well, today I want to talk to you about monarch tagging, and we're going to get into a little science here. We've been doing this monarch tagging for a very long time. We started the program in 1992 as a kind of on a whim, sort of. I had been working on African bees for a long time. I knew that money was going to dry up. Things happen in science, and if you're looking ahead, you can kind of predict where the money's going to go. I saw for the future that money was going to go into molecular genetics involving honeybees. I was not a molecular geneticist, and it would have taken me about 6 years of going back to school, in effect, to catch up with that field if I wanted to keep going with honeybee money. The kind of work that I did was demography and just basic biology of the bees and their relationship to everything else. And we had a lot of fun with that. We did some really interesting stuff with that because we worked on honeybee mating behavior, and the honeybee mating behavior things that we worked out turned out to be very instructive about honeybee mating in general. So it had a general application afterwards, but it's clear that that was going to change. So I was looking for something else, started this monarch tagging program, and we started the program with a press release that we released to the northern states because we knew the migrations would start in late August, early September. That press release was published on the front page of the Des Moines Register, and we were asking for volunteers. Within days, we had 500 calls back with people willing to tag monarch butterflies. And I go, wow. I mean, and at that time we were taking all the calls off an answering machine. So we were spending hours and hours recording all of these addresses, names and addresses where we were supposed to send tags. I mean, we were just overwhelmed. But we knew we had something. And so we knew also that the migration would be moving through Texas at a certain time. So we said, all right, let's send out another press release down south to Texas. Well, it appeared on the in the Dallas newspaper. Another 500 responses. So within the first year, we knew we had a tremendous amount of interest. And we proceeded to tag monarch butterflies with these people. It was really kind of curious how we managed to do it. I had no idea how to print the tags. And I was working with Brad Williamson, who was here in Lawrence, Kansas. He's a retired high school science teacher. He also worked at the university. And Brad knew how to print really small labels. And the idea was to come up with a printed label that duplicated the labels that Fred Urquhart at the University of Toronto had been using. Now, those labels he designed, were rectangular labels, very, very small print. And what you had to do with them is rub the scales off the leading edge of the forewing of the butterfly and the underside of the same area, and then make a little V of that tag and kind of hope it sticks on those two bare spots of the membrane. And we tried that the first year. And I got so annoyed with that procedure because I realized I had to change that. We couldn't be using those type of tags. First of all, one side would flip up and the other side would stick. And the butterfly flapping, pretty soon the tag is lost. Or worse yet, somebody breaks the wing in the process of trying to take off the scales. So I said, I'm not going to do that anymore. If we're going to go into the second year of this, we've got to come up with another tag. Well, we weren't very smart about how to do that either, but we gave it a try. We produced a rectangular tag with a set of glue that we sent out, new glue kits, in which we could affix this rectangular tag to the mitten-shaped cell on the underside of the butterfly's wing. And the problem with that was that people didn't know what a tiny bit of glue meant. So they were complaining about butterflies being stuck to the grass or being stuck to their fingers. And I said, please, just follow the instructions, a tiny bit of glue. Well, you know how people follow instructions. I know how I follow instructions, and I'm not very good at it either. But the point was that we had a problem, but I didn't know how to fix it. And we went for the next several years using those rectangular tags. And some of them actually made it to Mexico. And some people learned to tag pretty well, and that was fine. We were getting results, and that's what we needed. But I knew I needed a better tagging system. So I contacted 3M, and we said, we need an adhesive. We need a special adhesive that is fairly thick, put on a circular tag that we can put on the underside of the wing of the butterfly. And a tag in which we can have printed. So every tag is unique, has a unique code. And so that unique code could be identified with a person that did the tagging. And therefore we could get the kind of data we needed. So we had to contact a label producing company in St. Louis, and 3M, which came up with a really nice adhesive. And in 1997, we produced a circular tag, which was great. It was easy to apply. You simply peeled it off a backing and you put it in that discal cell of the wing, put your thumb on it for about 2 seconds. So you press the adhesive through the scales right to the membrane, and voila, it stayed on. And we weren't breaking any wings. We weren't gluing the butterflies to the grass. We weren't gluing butterflies to fingers and things of that sort. And all of a sudden, the number of tags that we were getting back recovered in Mexico went just through the ceiling. In the previous way of tagging butterflies, the recovery rate was about 1 in 5,000. When we got the new tagging system, we were down to 1 in 100. So it really made a difference. So that's an invention. I'm going to take credit for it. All right. So that was fun, and it turned out to be very successful. And over the years from 1997, well, actually from even the start of the program, although the numbers are small, we've tagged over 2 million butterflies, with about over 20,000 recovered in Mexico. And there have been a lot of recoveries in the United States as well, but most of them are like, oh, you tagged it over here and we found it on a playground over there, about a mile away or something like that. So most of the domestic recoveries haven't been very useful, but there's maybe 1% of those that are telling us something, and we're going to get to those eventually. But the other data has been so voluminous and so instructive and so enlightening about how this monarch functions during the migration that we have produced some really interesting data and really interesting papers about how this whole system functions, which was the objective in the absolute beginning. Now, I must say that in all honesty, I didn't predict all of the different types of things that would come out of this study. We set out, of course, to find out where the monarchs came from that get to the overwintering sites in Mexico. But beyond that, we want to know such things as where are the monarchs more successful? What time during the migration does it make a difference if they're early or late? Does it make a difference whether they take one pathway or another? Does it make a difference if they're riding down a coast or along a pathway that's across a lake or something of that sort in terms of whether they actually get to Mexico? So there are a lot of different things here. Are there differences among the sexes and so on and so forth? So we had a lot of questions, but the questions started to expand as the data started to come in. I mean, that's a sign that you're really onto something. So what I'm going to do is kind of give you a quick summary of a lot of the data that came through with this tagging program. But what I'm going to tell you is based on an analysis of data from 2004 to 2015, because that was the first database that we put together. The reason we started with 2004 is that in 2004, we started to ask people if they were rearing the monarchs that they were tagging, on the supposition that maybe, just maybe, the reared monarchs weren't doing as well as the wild monarchs. So we needed to be able to parse that out. So we did that from 2004 to 2015. And now we've got to do the task of analyzing the rest of the data from 2016 to the present. And it's going to be different, I can tell you that. The results are going to be different. We're going to see the effect of the increasing temperatures. The September temperatures have been increasing significantly over the last decade. And that makes a difference for the first phase of the migration. If it's too hot, the butterflies don't migrate. They don't like to migrate, and they don't migrate when the temperatures are in the 90s. They'll sit in the trees along the river someplace. And we know where to find them when it's in the 90s. They're going to be hanging out in the trees. They're not going to be, they may go at a flower or two, but it's going to be a brief thing. They're going to be hanging up in the trees somehow. They're trying to stay cool. They've got to keep a cool head for a very specific reason. They have to stay non-reproductive. This migration involves butterflies that are moving to Mexico that are turned off reproductively. Their whole behavior is designed to get them to Mexico. They don't need to spend a lot of energy producing offspring that will never get to Mexico. If they're reproductive at that time of the migration, their offspring are not going to get anywhere, and they're not going to get to Mexico. So these butterflies that are migrating are all non-reproductive, but they have to stay that way. And if you get 90-degree temperatures for a week or more, a lot of them are going to drop out of the migration. And the reason they're going to drop out of the migration is that physiology, that hormonal production is temperature dependent. So if the temperatures are really high for a long time, and we know this from lab experiments, basically the endocrine system that produces the juvenile hormone that sets the reproduction in place and in motion gets turned on if those temperatures are too high. So they have to stay cool, keep a cool head, because all of that endocrine production is in a couple of small glands in the brain itself. So we're looking at different conditions now, and hopefully we'll be able to talk about climate change and what's happening with the butterflies as we go on here. But I'm taking an especially long time to lay some background here for you so that you get an idea of how this whole thing works. The biology of the butterflies is important in this migration. They have to be non-reproductive. They have to have received the right information to become non-reproductive. So what does that mean? They don't just automatically become non-reproductive, just as they're born. If they're born when it's too hot, they'll become reproductive. So we don't fully understand the conditions that allow them to become non-reproductive, but there's a very sensitive period for the first 3 days after they emerge from a chrysalis. If they emerge from the chrysalis for 3 or 4 days and the temperatures are hot, they're going to become reproductive. If they emerge in the first 3 or 4 days, the temperatures, overnight temperatures are low, the daytime temperatures aren't too high, they will become non-reproductive. And part of that is associated with the declining day length. But the whole thing is kind of difficult to get at experimentally, so we don't have all the details yet. But it looks like the difference between day and night temperatures is really important in being non-reproductive, becoming non-reproductive. So there's still a lot of questions that we need to answer. And those are going to take some really interesting experiments and quite a bit of time to work out. So let's get to the data. Let's show you how this whole system works. Oops. Got to figure out which.
You just may have gone to sleep a little bit here.
There we go. Well, there's my circular tag. Isn't that a beauty? And every one of those has got a specific code on them. And of course, you run out of codes. When you're working with 2 million tagged butterflies over the years, you've got to do a lot of fancy things to keep every butterfly with a unique code for 32 years. But anyway, there seems to be an infinite number of possibilities, and we're still working through those. All right. This is the recovery rate for all sectors with more than 500 butterflies tagged. So what we did here is take a look at the continent of the continental United States from east of the Rockies, and we divided that into 5 by 5 longitudinal and latitudinal squares or rectangles here. They look like rectangles, but they're really squares. And then we looked at all of the butterflies tagged in those rectangles, and all of the butterflies recovered. And you can see here that the recovery rate from, if you look at minus 100 here, which is in the middle of the Dakotas, and you go straight down there through Texas, you can see that the recovery rate for that longitudinal swath is pretty good. But then if you go over here to the East Coast and look at the recovery rate, it's really low along the East Coast. And the relationships here are, I'll go through those in the next slide, but you can see generally that the recovery rate goes down the further east you go. All right. So, and if you look at the Southeast, there's virtually no recoveries out of the Southeast. So that's curious, but we don't have a lot of butterflies actually tagged in the Southeast, and we don't actually have a lot of butterflies that originate from the Southeast. That is, they're born there. So we're not quite sure what's happening in Georgia and South Carolina, but the number of butterflies that pass through there that are tagged are never seen again, except occasionally. All right. Well, if we look at the numbers tagged and recovered per sector, let's see, I'll get my picture out of there. Can we move this? Anyway, I don't know how to move my. don't need to see myself.
Uh-huh. There we go. Just put it down. here at the
Yeah,
bottom,
that's,
is
yeah,
that
yeah,
okay?
but yeah, put me in the bottom. I don't.
Okay.
Oh, that's wonderful.
There we
All right, thank you. All right, so here you look at the numbers tagged here. We had over 420,000 and recovered. We had about 3,800. And you can see how that shapes out. The numbers, the sectors are numbered in red. The number tagged is numbered in black, and then the number recovered is numbered is in blue. And you can see how that all works out. And you can see how the major tagging is going on in the middle part of the country. We have a lot of tagging going as far east as, say, Ohio, but it drops off pretty fast after that. All right.
Chip, is there a reason why there's nothing west of the Rockies?
Yeah, because of the population that we're working with migrates east of the Rockies. The population west of the Rockies migrates to the West Coast. All right, I should have explained that in the early pun. But so there's an eastern and western monarch population. And the western population is really much more mysterious, much less studied than the eastern population because the areas of the west are so vast. There's so many areas in the west where there are very few people, where the butterflies are breeding, that we really don't understand the whole breeding range very well. And the climates in the west are so variable from year to year that in some years we see butterflies coming down to Washington state pretty well and colonizing the California coast. In other years, because of weather conditions in the spring or during the summer, hardly anything comes out of Washington state. So it's the dynamic in the West is very different from the dynamic in the East. In the East, they all funnel down through Texas. They have to go through Texas, and they go through an area, about 90% of them, as far as we know, go through a place around Del Rio and Eagle Pass. That's, it's right on the border. So they're not going through, well, some are coming down through Corpus Christi and going through Brownsville, but that number is very small. So most of them are actually to the west of there because it's cooler. It's again, that heat factor determines the migratory pathways to some extent. All right, this is not moving forward again. There we go. Okay, recovery rates for latitudinal swaths. The recovery rate decreases from west to east, and the recovery rate decreases with distance. That is, the further they have to go, the less we see of them later on. Lateness of migrations increases from west to east. In other words, in the east part of the country, the temperatures are warmer during the fall, and that means that the migrations are later in the fall, and we'll get to what that means in a moment. As lateness increases, recovery rate decreases. Lower recovery rates east of 80 west, that's about middle of or western Pennsylvania, are due to distance, lateness, and pathways. From 80 west to 65 west, that would be from western Pennsylvania through the Maritimes in Canada, recoveries decline sharply from north to south, with few recoveries south of 40 north. The 40 north would be about Philadelphia, especially in the southeast. That goes back to that Georgia thing that I was mentioning before. From 80 west to 100 west, which would be western Pennsylvania again to the middle of Dakotas, south of 40, recoveries both increase and decrease slightly depending on the sectors. So what I'm saying there is that in this central part of the country, there are fewer recoveries of butterflies tagged south of 40. And that means that's probably because there are relatively few butterflies actually produced south of 40. So where is 40 north? We're almost at 38 north. 40 north would be at St. Joseph. Most of this breeding population that gets to Mexico is actually originally butterflies that originate from north of, say, a line from St. Joseph to Philadelphia. which is interesting, isn't it? In other words, we have a question to answer. Why is it that that is really the home of most of the butterflies that get to Mexico? Because it's too hot further south for them to breed continuously during the summertime. Butterflies virtually disappear from Georgia through Texas in midsummer. It's too hot. And we see them disappear here when it's too hot. 2012, anybody remember 2012? What happened in that summer? 2012, how many days in Lawrence, Kansas was it 100 degrees in July of 2012? 18. 18 days, July 2012, 100 degrees, Lawrence, Kansas. We had no monarch butterflies that summer. They just disappeared. And we can get into the physiology of that, but there's a serious issue here in terms of heat tolerance and longevity and what we call realized fecundity. Butterflies don't live as long when it's that hot. They don't produce as many eggs when it's that hot. Their larvae might or might not be able to survive, but the reproductive rate goes way down. Eventually, the population collapses. So there's a lot of lessons there, which we won't get into. All right. So this tells you a little bit about distance and days. It looks like the butterflies are moving at about 27 miles per hour per day for weeks and weeks and weeks. And if you look at this and we go 45 north is Minneapolis, St. Paul. All right. Supposing we have a butterfly that starts at 45 north all across the northern part of the country. As you go east, that distance gets larger and larger. All right. So if you go out and calculate the number of days from 45 north Minneapolis, St. Paul, would take them about 41 days to get to the border with Texas and Del Rio and Eagle Pass. On the other hand, if they're 45 north and they're on Prince Edward Island in the Maritimes, it's going to take them about 83 days to get to that border if they survive it. So the problem that they have with increasing distance is pretty clear. There are a lot of hazards on the way, and to take that many more days, twice as many days to get to that border crossing is really a significant problem for them. And we can see that in the recovery data. Here, the blue line represents the distance. The red line represents the recovery rate. And then we have a linear recovery rate in there, a statistical plot. But what you can see is that most of the butterflies that are recovered, that get to Mexico, that are recovered in Mexico, originate from about 80 west, which is 90 west, which is just west of Madison, Wisconsin, all the way to 105 west, which is in western Dakotas. So this whole area to the west is really the population that's producing the butterflies that get to Mexico, as opposed to relatively small numbers from the east. Now that is a story that I don't want to tell a lot of people in the east because I still want them to tag. Because, you know, we don't give people expectations about their results because you don't want to. You want to have it be objective. So we tell them to tag as soon as they see butterflies in the first 10 days of August and right until the last time that they want to get out there and tag a butterfly. And so we have people even tagging butterflies in November, which have no chance of getting anywhere, but that's fine. That tells us because they're never recovered. And that the fact that they're never recovered is important to us. All right, now let's get into where I had a lot of fun with this. And this, I'm telling you a story that took about 20 years to sort out. When I got into this, I had no idea how the migration functioned. But after 2 or 3 years, it became clear that there was some orderliness to it. And at that time, I was working with a guy by the name of David Jabot, who was a glider pilot. And he was talking about all the conditions during glider piloting, the physical conditions, what you were looking for, what you depended on. And a lot of that was meteorological data. So I was concerned about this, and I was observing that the migration was arriving at certain points almost at the same time every year. So I finally called up David and I said, David, I need to have a website that tells me what the sun angle at solar noon is every day for every latitude and every longitude. Oh, that's easy. Yeah, sure. So right away after that, I looked up and it say St. Paul, Minneapolis. So altitude angle at solar noon when the butterflies first got there was about 56, 57, something like that. When they got to Lawrence, Kansas, it was about 56, 57, the leading edge. I call up Bill Cowart in Texas. I said, Bill, I've got a question for you. I want to know the leading edge of the monarchs when they get there, coming from the north. I want to know the date. Oh, that's easy. It's 26th, 27th of September. Okay. Okay, Bill, I got to tell you something. I know what the sun angle at that time is. And it's the same sun angle I'm seeing all across this migration. So what's going on here is that as the fall progresses, the sun angle at solar noon declines slightly. And what we eventually found out was that the monarch butterflies began leaving at the northernmost area, coincident with the sun angle starting to reach 57, 56, 55. So at Winnipeg, they're starting to leave at the end of the first week of August. Wow, they're leaving early. So they've got a long way to go, and they're leaving early. And it's coincident with the sun angle at solar noon. Part of the thing that got me interested in all of this was the fact that if we look at what happens in Mexico, we know about the Day of the Dead, right? And we know that they celebrate the arrival of monarchs coincident with the Day of the Dead. Oh, there's a clue. So what is the solar angle when the butterflies arrive in Mexico just prior to the Day of the Dead? 57. Damn. But how can we prove it? How can we get the data that actually shows the linkage between one and another? You have to do a lot of tagging. And you have to have decades of tagging data because there's a pattern here and it's sloppy because butterflies are tagged before the migration and butterflies are tagged way late in the migration. So we need to sort all of that out. Wow. There's a migration window. That blue represents all the butterflies that get to Mexico. It's a migration window. If you are tagged before the migration reaches your latitude, you still have a good chance of getting to Mexico. If you are tagged after that migration window is essentially past 47 degrees sun angle at solar noon, your chance of getting to Mexico drops really fast. So probabilities are that, and this is that 18 years of data, or no, 11 years of data. Chances are what you're looking at here is really an opportunity. And if the butterflies are not in that opportunity to get to Mexico, they have a very low chance of being successful. Now we have a lot of data here. All of those red butterflies who were tagged, and there are literally tens of thousands of them, did not get to Mexico. And what do they represent? They represent small butterflies. They represent butterflies that emerged late. They represent a lot of small females. There's a sex ratio changes during this migration as well. So this is really, I could go on about this, but this migration window has been interesting because what we're going to see in the next set of data that we're going to analyze is that the ability to get to Mexico is defined by temperature in September. And there's going to be a lot of that red to the right of that curve, a lot more of that red, because a lot of butterflies are going to be on the move late. All right, I'm not expecting you to see anything here so I'm going to explain all of this later. But what we did was put together the number tagged and recovered in Mexico by longitude for 98 through 15. And the longitudinal values are to be read vertically, and the other values were horizontally. So this takes hours to stare at. And I've done that staring. I'm going to help you out here. All right. So the effects of the loss of habitat on tag recoveries are interesting. There are two intervals in this whole history. First, a period during which common milkweed was widespread in row crops, that is corn and soybeans, followed by a period after 2005, during which milkweed all but disappeared in crops due to the adoption of herbicide-tolerant crop lines. We're talking about glyphosate, we're talking about Roundup. They started using these lines of crops that were genetically modified to resist glyphosate, and that took out milkweeds. We could talk more about that later. The numbers tagged each year in the latter period declined sharply in the upper Midwest. In other words, you take out the milkweeds in corn and soybeans, the number of tags that people applied to butterflies because the population was much lower because there was no breeding going on in corn and soybeans anymore, that declined. So this was a clear indication of the impact of the use of glyphosate on the loss of habitat. Habitat declined. I could show you a lot of pictures of corn and soybeans with milkweeds growing in them. You can't find milkweeds growing in corn and soybeans anymore. And there was a study that was done in about 2000 when we were first looking at this problem that showed that probably the major portion of the butterfly population at that time, prior to 2000, was actually produced in corn and soybean fields. And we took that away from them with that technology, and the tagging went way down. Well, there's more details here. We won't go into that. In the Northeast, there were 5 years in which the percentage recovered per longitude in Mexico was 0.12 or higher. And this increase was associated with a decline of milkweeds following the adoption of herbicide-tolerant things in the Midwest. So as the Midwest went down, the Northeast went up in terms of the proportion of butterflies that we were covering in Mexico. And that's as you would expect. We see the same effect if we have a drought in the Midwest and really good conditions in the Northeast. The proportion getting to Mexico is a function of that balance. All right, let's talk about migratory success. And that was all in that graph. Migratory success declined in years with droughts, 2000 and 2011, extreme high summer temperatures and droughts in the Midwest, 2012, extreme low summer temperatures. You may not remember this, but 2004 was really cool. And 2009 was cool as well because the jet stream was way up there in Canada. Or actually the jet stream had come way down, I'm sorry. It's just the opposite. When the jet stream comes down, it brings cooler air down into the jet stream goes way upwards, and it's going to go way up in the future. It's going to get a lot hotter here. All right, delayed migrations due to September and October temperatures are associated with declines. Late recolonization. So what I'm talking about there, late recolonization means the butterflies coming back from Mexico are late, and they are late in recolonizing the area north of 40. Then that results ultimately in a decline in the overwintering number of butterflies. So there are multiple negative factors in several years. And there are a couple of years I haven't been able to figure out yet. So there's still some work to do. The recovery rates are highest every time we see a year in which everything is close to the long-term average. If you think about it, the butterfly is really adapted to long-term averages, right? And that's the way all things are. they're not adapted to extremes. They don't occur often enough. If they do occur often enough, then there is a slight adaptation to an extreme. But basically, evolution is not predictive, right? The butterflies can't predict evolution. They have to respond to the changing conditions. So their representation is always very close in terms of adaptations to the average conditions. And when we see average conditions, we can predict the population is going to be as big as possible and that we're not going to see any declines associated with any of those other factors. All right, there are other factors involved in migratory success, pathway length. You got long pathways along the East Coast. They actually come down, they go along the Gulf Coast as well. So the butterflies are coming down the East Coast, then they swing over through Georgia and they come along all the way through Corpus Christi and into that Del Rio Eagle Pass area. That's a heck of a long route, and it takes them about 2 weeks longer to travel that route than it does to go right straight down from Minneapolis, St. Paul, right across the border. And we have reared versus wild, and what we've shown with reared versus wild, which I don't really want to get into in detail, but the reared butterflies have a hard time making it to Mexico. And there's a whole bunch of reasons for that. People tag everything. Even something that's got 3 wings, they'll put a tag on it. Oh, we hope they'll get there. Well, that takes the numbers down because they're not going to get there. All right. So you got to, if you're going to have butterfly that gets to Mexico, it's got to be fit. It's got to be large. It's got to be disease-free. It's got to be attuned to all of the environmental conditions that allow it to get into a non-reproductive condition. And we don't know all of that. And the breeders don't know all of that. And some of them are rearing them in houses where they don't have access to environmental cues. So we got a lot of things going on there. Furthermore, a lot of that rearing goes on in the Northeast where the butterflies have a hard time getting to Mexico anyway. And we're looking at a wild population that is kind of pre-selected. If the wild butterflies aren't fit, they're not even going to be tagged because nobody's going to see them. They're only going to die or disappear before they're actually tagged. So they're, you know, the wild versus reared is kind of a catch-all for a lot of different sort of things to analyze and think about here, but we've got to get people who are doing the rearing to do a little better job of it. All right, migratory success number 3, timing is important. This migration window is important. If you're late to the party, you're not going to get to Mexico. Location, the latitude and longitude, as I explained before, size and sex. Who gets to Mexico the best? Yeah, the males, the big machos, the ones with big wings. The big wings, they've got better lift with those big wings. They'll get carried a little bit better on the winds. Yeah, and their forward speed is actually better. So their glide ratio is better. Their drop rate is less. If you got big wings out there and you're a hang glider or something like that, your drop rate, it can be really quite low. I mean, this is all about gliders, right? In effect, the Monarch is a glider going to Mexico in some ways. And so its drop rate is a function of its size. And the females have a faster drop rate because they're smaller. So they have a harder time actually getting to Mexico. And by the time you look at Mexico, 55% of the arrivees are males, 45% are females. Now the females get their due on this because they live a little bit longer. And the males get randy pretty early in the season, and the mating goes on with the small males at the overwintering sites early in February, and they breed themselves to death and die there, whereas the females trot on north. All right, well, we have other things, drought, weather delayed migrations, droughts in Texas and Oklahoma are really a problem. We had 2 years ago, we had a tremendous drought in Mexico, and the population was extremely low. And it turns out that when we looked at the tagging data, we could see that we tagged a lot of butterflies in 2023, but they didn't get to Mexico. And the reason they didn't get to Mexico is that they were going through droughts from Oklahoma almost to the overwintering sites. Really incredibly severe droughts. So that took a lot out of them. And we've done a fat body study. The butterflies are living on stored fats a lot of the time. And they're converting these stored fats into sugars. There's a sugar called trehalose in insects that fuels the metabolic processes. And to get that trehalose, they have to actually get a lot of nectar first. The nectar is converted to fats, then the fats are converted to the trehalose. That gets them through the winter, that gets them through the migration, and so on and so forth. So there are other factors involved here, but we don't need to really get into them. So let's figure out, this is the first. We did this a couple of months ago. One of the things we've wondered about for a very long time is how many of these butterflies that start a migration actually get to Mexico. So how do you figure that out? We've struggled to try to figure this out. Well, we know how many butterflies are tagged. We know how many butterfly tags get to Mexico. We can also estimate the number of tags that we don't recover in Mexico that are still on live butterflies. And we can estimate the number of tags that are not recovered at all or accounted for on the basis of the existing butterflies. Make a lot of guesses, in other words. And those guesses are based a little bit on the size of the overwintering population and what we know about how many are tagged. Turns out that the numbers that are tagged each fall is pretty well closely correlated with the number of butterflies that reach migration, except in those years like 2023 when we have bad droughts. So we've actually said that under favorable conditions, only about 15% of the butterflies that start the migration actually get to Mexico. When we get conditions like 2023, it may be only 1 in 20. Now that seems low, and maybe it is a little bit low, but it could actually be high. We still have a lot of work to do, but for the very first time, we've got a ballpark idea of what it takes to get to Mexico. And most of those butterflies that are getting to Mexico are coming out of the Midwest, the upper Midwest, Minnesota, Eastern Dakotas, Western Wisconsin, that area. That's the area that's producing most of the butterflies. About 71% of the butterflies that get to Mexico come out of that region, all north of 40. So we're making progress. We've got more work to do, but right now it looks like we're zeroing in on a lot of the factors that are really important in trying to explain this migration. Now, I don't know what time it is, but I promised you guys that I was going to talk a little bit about climate change and conservation. You all